All right. So yeah, my name's Brian. Today I'll talk about Bayesian stable isotope mixing models, and specifically this MixSIAR R package that I've worked on mostly from 2013 to 2016, but then with a couple updates in 2018 and 2020. So I have to uh, admit I'm not up to date in what the developments have been in the last few years, but hopefully this will be a good place to get started because I think it is currently used by a lot of people still. So yeah, so the goal of these mixing models is to calculate the percentages of different sources to a mixture, and they're used in a few different ways in different fields. The canonical example that we keep using in this MixSIAR package is the diet of wolves, which are eating three sources: marine mammals, which is about the cutest picture of a seal I could find at some point, salmon, and deer. Yeah, so this is the MixSIAR wolves example that you can find a vignette on that web page, and I'll go through at the end. It's from a 2009 paper. People have also used these types of models to calculate movement of seabirds from different colonies, where I think the mixture was the juvenile birds, and the sources were stable isotope signatures from the adult nesting birds in different colonies. They're also used by soil scientists to calculate the percentages of different soil types to a river in a river system where they collect sediment as the mixture. But mostly, or at least they started out in trophic ecology, where we're trying to estimate diet, and so the source proportions are the goal, kind of the main focus that those are the parameters we're trying to estimate. So in this wolves example, the result from the model might be that these wolves are eating 10% marine mammals, 20% salmon, and 70% deer. So those percentages are what we're after. And maybe the answer is 35% deer, 20% marine mammals, or 45% salmon. And then these mixing models are using biotracer data. They can be different types of tracers. They're most commonly, or they originated from stable isotopes. And the basic idea is that the consumer or the mixture is somewhere in between the three different sources, and the relative position of the consumer stable isotope values indicates the percent of the diet, because the carbon and the nitrogen that is in the consumer's tissue originated as one of those sources. So if you take stable isotope values of the source prey items and of the consumer that is eating more salmon will look more like salmon in this kind of isotope biplot. And if it's eating more deer, then the wolves will be closer to the deer. So that is the type of data that we fit these models to calculate these diet percentages. And they start off quite simple. We can start with a linear mixing model. Let's consider that you only have one stable isotope, delta 13 carbon. So your consumer was negative 27, source one negative 28, and source two negative 25. And then we're trying to estimate what was the diet of that consumer. The model equation, there's two equations, two unknowns. We want proportion source one, proportion source two. And it's simply the proportion source one times the value for source one plus the proportion source two times the value for source two. And then we have this sum to one constraint, which makes these models somewhat different. The proportions have to add up to one. So we have two equations, two unknowns, and we can solve. So the consumer is basically twice as close to source one as it is to source two. And so the proportion of source one is twice that of source two. So that's the basic idea. Quickly can get more complicated. So if you have Now we have, in addition to carbon, we have a second tracer. We have delta 15N. So now we can add a third source. And now we want the proportion source 1, proportion source 2, proportion source 3. We have now 3 equations, one for the carbon, one for nitrogen, and then the sum to 1 constraint. But we can solve that. Where linear mixing models have issues is once we start incorporating more uncertainty. So there's not just one value for the consumers, there's not one value for the sources. So how do we take into account this uncertainty? Maybe we have additional sources. So now we have 4 unknowns and only 3 equations. Maybe we have a 5th source. So we have 5 unknowns and only 3 equations. So that's why starting in 2008 or so, people gravitated towards putting these models in a Bayesian framework, which accounts for the uncertainty in the source values and the consumer values, and then you can fit all of the data in a hierarchical Bayesian framework and take into account this uncertainty in a nice way. One reason, so the result of that is a posterior density estimate for each of the proportions. So these are typical output plots from MixSIAR or another Bayesian mixing model you might use. So in this case, we have 3 sources, brown, green, and red algae, I think. And so this is a posterior estimate. You have not only do you have the mean or the mode or the median estimate, but you also have the full posterior, which gives you a credible interval. One reason why we don't like to use maximum likelihood for these types of models is that they're often, it's having the full posterior is nice because you may end up with multiple possible solutions. And in that case, you end up with these bimodal posterior distributions. And if you had used maximum likelihood, then you may get stuck on this alternative solution where it's the model saying could be 100% the green source and 0% red and blue. Or there's also a substantial likelihood that it could be like 50-50 green and red. So if you don't get the full posterior, you just use the maximum likelihood, then you could end up into issues here. The idea behind this MixSIAR package is it incorporates several years of methods development that are building on each other. So this first one in 2001, there was a model called ISO-AIR that it incorporated in an ad hoc way some uncertainty in these proportion estimates. And then the next year, Don Phillips has published a bunch of these methods improvements, added concentration dependence, which is admitting that the percent of carbon and nitrogen may differ between the various sources. And that kind of warps the mixing polygon. In 2003, they added this, they came up with isosource. In 2004, this was the first Bayesian model called MixSIAR. It was written in MATLAB. It had a GUI. Close on the heels of that, there was a MixSIAR model that you may have heard of and used. It added a residual error term. It was written in R, which was nice. In 2010, people added the ability to fit the source data hierarchically. And then people have also added improvements like including covariance between isotopes because the isotopes typically covary. And then covariate effects have been added as well. So a good overview of all of these kind of individual iterative improvements is this paper led by Don Phillips in 2014, and there's the link there. So the general idea behind the package is that we use R to load in the data, and then you choose the model options. And then there's this function write JAGS model, which will write a JAGS model file based on the structure of the data and the options that you'd like to include. So MixSIAR is not one model, but it's a framework that sets up a mixing model according to your data and the types of things you'd like to include. And then we run the model in JAGs, and then all of that comes back to R, and we can look at the diagnostics and output. So the plan for today, I already gave you the introduction. The other things I'd like to talk about are source fitting and covariate effects, because I don't think that the other mixing model software includes those. I could be wrong. Another package that's currently being developed, though, is SimMer. That's one to check out. And then effects of priors, because sometimes that can be misinterpreted. And then I'll share the GitHub site and some resources. So when I say source fitting, that is simply taking into account that there's sampling error of the sources. So we don't know the true population mean and variance of the sources. We just have a sample. So we fit a hierarchical model that allows the source means to deviate from the sample means of the data that we have collected. And this can be done in a couple ways in MixSIAR. If you have the raw source data, then that is done if you can find the equations here in this Parnell et al. Paper from 2013. If you have the raw data, that allows us to include the covariance between, say, carbon and nitrogen isotopes. If you just have the mean and standard deviation and sample size of the source data, then we can still allow the means to wiggle around to maximize the likelihood of the mixture data, but in a way that assumes that the isotopes are independent, because we don't have the raw data that would have the covariance information. And then if you don't want this behavior, or if you like to see how sensitive the results are to this behavior, you can artificially set the sample size to a really high value, like 10,000 or something. And then that won't allow the source means to move at all. So the effect is, say we have two isotopes, and we have three sources, and these are our mixture consumer points. And these are the sample means. If you allow the sample means to not be if you allow the source means in the mixture likelihood to not be exactly at the sample means, that changes where the polygon is, where your mixture proportions will be calculated. So it depends on the sample size and the natural variance of the source data. So if you have you've done a lot of sampling and you really know where the sources are in the ISO space, then these won't move as much. And if you have more uncertainty, then they can move around to maximize the likelihood of the mixture data. And then one important point to note in this process is that in the mix equations, the variance of the sources and the trophic discrimination factor, or the trophic enrichment factor, is indistinguishable. So if you really don't know what those values are, then effectively you don't know where the sources are in the mixing polygon, and it adds uncertainty to your estimates of the proportions. But if you see these papers for a more thorough explanation. And then these are the three kind of options that MixSIAR has, and those are detailed. If you go to this paper is kind of where we outlined the MixSIAR model framework. And there's a supplement that describes in less technical academic speak, but more like helpful, straightforward speak, what the options are and how they work. All right, next section. Another motivation for writing the MixSIAR was to allow for some simple covariate effects. So it's important to note that if you're using MixSIAR or SIAR or any of the other models that were there before MixSIAR, you're just estimating the mean diet or the mean proportions of the consumer. Or effectively, you're assuming that all the consumers have the same diet. There's no variability based on group membership or body size or anything else that you might have information on. So you're only estimating this mean from all of your, if you have 20 wolves, say. But there's a few different ways we can include extra information and allow the diet estimates, the P's, to vary by group membership, categorical covariates, or linear covariates. And what we do is the issue is that we have that sum to one constraint. So these proportions are what are called compositional data, or they're not independent. So we transform the P. We have a Dirichlet prior on the proportions. This is a bold because it's a vector, because there's, say, if you have three sources, you have three proportions you're estimating. If you do a transformation, we use something called the ILR transformation, then we can include effectively a linear regression in this ILR transform space. So we can estimate like a global mean. We can estimate offsets for membership in the different factors, or we can estimate like a slope term that could. The example that I will show is for alligators of different lengths, then their diet estimates change as a smooth function of length. Oh, yeah. And there's a couple different ways to do the categorical effects. You can either treat it as a fixed effect or as a random effect that I'll show you. So how the fixed effects work, instead of assuming that all consumers are the same and just estimating a mean diet, this is the simplest way to do it, is to estimate a mean diet proportion for the different groups independently. And this is similar to running a separate model for each group, which you can do in other software. But one difference is that the source data, I said that was hierarchically fit. So that is only being done once. Whereas if you fit separate mixing models on each mixture or consumer group, then you would let the source means be moving around in a different way if you're running them independently. And then there's also error terms in this type of model that would be fit one time and shared across groups. So there's some not obvious differences at first. The other way that you can include groups or categorical effects is to assume that all wolves are similar, but they have these different groups that there's some variation, but their group mean is randomly distributed, centered on the overall population mean with some variance. So we estimate if you have a lot of groups, this estimates fewer parameters. So that's a pro. You need at least two groups in order to estimate this variance of the groups in order to do this. But this is a nice way to account for variation from some kind of categorical factor that you can measure if you have a lot of them, but without estimating so many parameters. And then you can also, these can be nested or hierarchical or not. So that 2009 Wolves paper that we use as the basis for this example we had an overall or global proportion of deer in the diet, say, and then we have variance that's estimated, allowing wolves in different regions to have different diets. So then we have a proportion of deer in wolves from region 1, region 2, region 3 that are estimated. And then there were also, they knew that the wolves were in different packs. And so the pack was nested within region. So the estimate of the diet of wolves in different packs, say packs 4 to 7, were in region 2. So you took the overall kind of deer proportion estimate, and there's an offset for region, and then there's an offset for pack. And then the individual diet estimates were from deviations from the pack mean. So you can do this. You can either say that these, if you have multiple effects, you can say that they're independent or they're hierarchical, and that is just the only difference is that when you say the pack, you want the estimate of pack 4, it either will just take the global estimate plus the deviation for that level of that factor, or it will also add the offset for the region that pack is a member of. And this is a picture of the GUI that MixSIAR used to have, but doesn't work anymore. So the end result of that type of nested random effects analysis is that you get these proportions of each source, but they're by region and also by pack, say. All right, so that was categorical or fixed or random effects. The other type of effect you can include in MixSIAR is a continuous effect, and that is simply a linear slope term in ILR space. So that the reason why it's not a line is just because of the ILR transform effect. But the example that I'll show is from alligators, that there was only 2 sources. It was freshwater and marine, and they small alligators are mostly eating food sourced from freshwater, and as they grow bigger, they in a continuous fashion, they gradually shift their diet towards marine-based sources. So those data are from Nifong et al. And then we show a reanalysis in that MixSIAR paper showing how you could do it with a continuous effect instead. And then there are, if you are willing and able to code your own JAGS models, you could add a lot more complexity that is beyond the scope of a package. It's kind of difficult to write in a general way all of the things that you might want to do. But this is just an example of a pretty cool, quite complex effect from the Parnell paper, where they had this multivariate spline that allowed their source means to be time varying and also have covariance. And then the diet proportions were also a smooth function of time. That's just quite neat and I think worth highlighting. Okay, next section is on the effect of priors. So these are Bayesian models. So all of the parameters need to have priors. This was some of this material is from Michael Brett 2014 paper, and then also discussed in the MixSIAR 2018 paper. So because the proportions are not independent, the priors it's not intuitive or really obvious that there is an uninformative prior, which people sometimes want to have in a Bayesian analysis. So we have these Dirichlet alpha parameters, and setting each of them, if we have this example with 3 sources, we have 3 proportions. If we set the alpha equal to 1 for each, then the prior looks like this. So it's not really flat. If you're just looking at the marginal distribution for the proportion of source 1. But the reason why this alpha 111 is considered the uninformative prior is that if you look at the combination of all possible P1 and P2 at the same time, any combination is equally likely. And so for that reason, for diet example, sometimes we call it the generalist prior. So it's uninformative, or it's not really uninformative. It is assuming, the prior information is that the diet is generalist. All proportion combinations are equally likely. So it's uninformative on the simplex, or this combination of proportion space. You might think that something else that looked prior that looked like this, alpha 1/3, 1/3, 1/3, looks more uniform, perhaps. But that actually puts extreme weight on, it puts heavy weight on the extreme values of very low or very high proportions of source one and source two. So it's not less informative than the alpha 111 prior. Then yeah, so what is the effect of this? It depends on how much data you have and how informative it is or how good it is. If you have this situation where you know the sources quite precisely, it'll have less effect. If you know you don't really know where the source means actually are, then it'll have more effect. And so Brett showed this where if you have this situation where you don't know where the sources are really, it'll estimate, even if the truth is that the consumer is quite close to this source up here, the truth is 80% this source, your estimated posterior will be about the prior. It'll be about 1/3. And that's because you only have one data point and you don't know where the source means are very well. And so he calculated this surface area, a standardized or normalized surface area metric, where it's effectively the distance between all of the source means, but normalized by the uncertainty in the sources. So in here, you don't have as much information as you do over here. So even if you only have one data point here, if you know the source is quite precisely, or they're spread out in isospace in such a wide way as opposed to a really narrow triangle, then you have more information to estimate the proportions and you can get the answer right. So the consumer sample size invariance will affect the impact of the prior. The source sample size invariance will affect the impact of the prior. And then the source geometry, or how much space is between the source means, will also affect it. And that, if you're designing a study, you can control the sample size of the sources. You can control the sample size of the sources and the consumers, but you don't know in advance what the values, what your polygon is going to look like. So you could just kind of end up getting unlucky if the source geometry isn't favorable to estimating these models. And sometimes that is just the answer, and that's unfortunate. So here's an example of the source geometry. In this ISO space, we have quite high source uncertainty. So what could you do to minimize the impact of this, of having a prior? You could collect more samples. You could also, because the source and the discrimination factor variance is indistinguishable, you could do an experiment to try to get to lower this discrimination factor variance and reduce these error bars. Here's an example if you have too many sources. So if you only have two tracers, two isotopes, and now you try to run a mixing model, but you have 6 sources. One thing you can do is combine. You can run the model with 6, trying to estimate 6 proportions, and then you can combine those afterwards, or a posteriori. You can also try putting on informative priors if you have information from stomach contents or fecal contents or visual observations. You can also include more tracers, because sometimes that can help if you had, because it gives you more more data to fit the models too. So this mantis strip example is available as in the MixSIAR package, and it turned out that it was difficult to estimate a model with this many sources, but in the interesting ecological story here was not the proportions of each of these 6 individual sources, but the interesting thing was the relative proportion of the soft-bodied prey versus the hard-shelled prey, given the type of consumer, because it's a mantis strip that has this hammer, and the idea was that it's breaking up in hard-shelled prey, but we showed that it actually is eating quite a bit of soft-bodied prey, and it doesn't matter whether it's fish or brittle stars or whatever these were, I forget. But it matters that it's this collection of soft-bodied prey that was interesting. So that's sometimes that can help. Sometimes if you have this, and this is the killer whale example that's also included in the package. You have sources that are confounded. So what do I mean by that? You have these are your killer whale consumer points, and then you have kind of two sources that are behind each other. And so the model can't tell whether it's a Chinook salmon or coho salmon that should be bringing the consumers up to the upper right. And in that case, an informative prior from other information can be quite helpful. So the killer whale examples shows one way how to do that. Yeah, so if an informative prior seems like it could be a good idea, there's a couple things to note about how to go about constructing them. And one thing is that you control not only the mean of the prior, but also the variance or the informativeness of the prior. So this is our alpha 111 quote uninformative prior. If you have alpha 10 10 10 or 100 100 100, all of these are still centered on 1/3 of each of the three sources, but they're tightening it down and making it more informative, such that if you use this prior, you would need a lot of data to pull the model to pull the posterior estimates away from the prior. So let's say that this is an example from I think this is the wolves, but you had 30 of source 1, 8 of source 2, and 25 of source 3 in stomach contents from consumers. You could construct a prior using those raw stomach content values as the alphas, but that's pretty informative. So the sum of the alpha is 63 in this case. So that's actually quite a bit more informative than the sum of 111 is only 3. So our suggestion as a starting place is to scale those by the number of sources so that the sum of the alphas ends up being 3, the number of sources, and then you end up with something that looks more like this. So this source is more likely, but it's not, you're not telling the model that these solutions are totally impossible. Okay. Those are just a few kind of topics that I thought were worth highlighting. If we now want to go and get MixSIAR, it's available on CRAN. It's also available on GitHub. Both of them require you to install R and JAGs, and then you can install from the typical way with install.packages, or you can install using install GitHub. This is the one that I would recommend because it gets the latest updates. And I don't think I have pushed a release to CRAN in a while. Other resources. There used to be a user interface, but it is no longer operational. I tried and I couldn't make it work the last week. It was unfortunate that I started this project right before Shiny came out, but that is life, and it's not worth making another one. So I now use the script version of MixSIAR. It works, and it'll help you learn how to use R, and everything will be reproducible because instead of clicking buttons, then you have the script, you have what data file you used and how you set up the model, and everything's right there. We wrote a pretty extensive manual, and this is available when you download and install the package, or you can go directly to it on GitHub, but it walks through in a much more, the explanations are much longer and more, if you're just starting out with isotope analysis, that I think would be helpful to walk through the manual, goes through the different working examples and then how to use MixSIAR on your own data. And some things like I said, the source and discrimination variants are indistinguishable. The model description, all the equations are in this PeerJ paper, and the supplement is also worth looking at. We've included, I think, 11 example data sets. Some of them have built vignettes, and some of them just have example scripts, but they're all included in the package install. There's also an issues page on GitHub, which This is a recent screenshot, and I apologize if you've asked me a question I haven't gotten to because there's just a lot of questions, and I only have so much time, and I haven't really been focused on isotopes lately. So, but at this point, there's a broad user community. So, advantage of posting to the issues page is that someone else may have had that same issue, and you can also search through past issues to hopefully get help if you need it. And this is just a kind of, it documents the conversation of reporting bugs and then getting them fixed, or cross-linking between issues. It's quite nice. So use the issues page if you have issues. So that is all I have for slides. Now I'm going to attempt some on-the-fly coding, which is dangerous, but hopefully it works. I am planning to go through a couple of the included vignettes that that come with MixSIAR. So here we are on the Statistical Methods seminar page. And if you go into the MixSIAR folder, you have a link to the MixSIAR site, the MixSIAR paper, where to get R, where to get JAGs. So I've assumed that you've been able to do that or will be able to do that in the future. And then we'll go through the wolves example. All right, so the wolves example. Let's see. All right, so we can, assuming that you've already installed MixSIAR, you can load it with library MixSIAR. And then if you want to if you want to find where all the example scripts are, this will show you. You can use find up package MixSIAR, and that will you go to in your R library typically, and then the version number, and then scroll through all the amazing packages you've installed, and eventually you'll find MixSIAR. And then in here, this is worth finding because it has the manual. And then it also has a folder of example scripts. And then, so you can open up each one of these. And then this, so the one that I am following will be the wolves example. There's also all of the consumer source and discrimination CSV files. So you can check what those files look like if you're trying to match the format with your own data. It could be helpful just to pull some of these open. So that is why we have this little bit of code to show you where that MixSIAR got installed to on your computer. Then let's see. Okay. We're going to pull up the wolves example. There's the diet of 66 wolves. We have 2 tracers, carbon and nitrogen. We're going to include 2 effects, region and pack, where pack is nested within region. And we only have the means and standard deviations of the source data. We don't have raw source data. So first thing to do after loading the package, point MixSIAR to where the consumer data file is, and then call load mix data. And we're going to say that the isotope names, these are the columns of the CSV file. These are the 2 factors. We're going to treat both of them as random effects and not fixed effects. If it was fixed effects, we'd say false. And then since region is this one, this second one is pack, we're going to say that pack is true. It's nested within region, and we don't have any continuous effects. So we have that. And now we have this object mix, which just has our isotope data. And it has the factor levels. So it has the region 1, 2, and 3 for each of our wolves. And then it has the pack membership for each of the wolves. And then it also has this factor to look up is which region is each of the packs in. So pack 1 is in region 1, pack 4 is in region 2, 5, 6, 7 all in region 2, and pack 8 is the only pack in region 3. And yeah, so that's our mixed data is loaded in. Now we have our source data. Just again, have to have the file name. And then this example, we actually have source data by region. So if you pull open the source, wolves source.csv, you'll see that each of the regions are, we have the deer, the salmon, and the marine mammals isotope values by region. So we can include region-specific source data. So let's load the source data, see what that looks like. We have the means for each of the 2 tracers by region. We have the standard deviation by region. We have, what else do we have? And we have the sample size by region and source. Then the third data file we need is the discrimination or the trophic enrichment factor. If you are a soil scientist or you don't know what trophic enrichment or trophic discrimination factors are, it is possible that those values could just be 0. But let's load that file. And in this case, we're adjusting the sources by 2.1 for carbon and 2.6 for nitrogen. Once we have those files loaded, we can make an isospace plot. Ooh, that's not good. It's strange because I was just everything worked yesterday. Anyway, so there's an error with the PDF saving, but here we still have the plot that we can look at now. So these are our wolves here. They're colored by region, and then the shape is the pack. Deer, salmon, marine mammals, and we have source data by region, so those are also colored by region. I talked about the source or the polygon geometry and why that matters, because you can see this is kind of like this mixing polygon is quite narrow, which effectively is reducing our 2 dimensions into a 1-dimensional problem and makes it more difficult to estimate the source proportion. So we've included this calculate convex hull area function, which Brett used in that paper. So we can calculate the convex hull area of our polygon, which is standardized by the variance. And we get, interestingly, so there's one calculation for each region because we have source data by region. The region 1 is really small compared to the other regions. So that if you look at these salmon or red colored, so that triangle is extremely skinny and effectively 1 dimension. So that can be an issue with estimating the source proportions in that case, because salmon and marine mammals are both, it could be a lot of deer and salmon, or it could be a lot of deer and marine mammals. It's difficult to estimate the salmon versus the marine mammal contribution to these wolves' diet. The blue triangle is a bit more, there's a lot more area compared to that salmon colored polygon. So it'll be easier to estimate the proportions of these 3 sources just because of the geometry of that mixing polygon. So it's useful to look at the convex hull area. Next thing that we like to do before running a model is to plot our prior. Always plot the prior and always look at the prior. In this case, we're just using the uninformative prior, which is the alpha all one. The next thing we need to do is choose the error option for the model error option. I didn't talk about that, but you can see this Stock and Simmons paper that describes the differences between those three. We're going to use the residual times process error option here, which is true and true. And then we're going to write, once we have the structure of our mixed data, our source data, and we have the covariate effects selected, then that is going to define the JAGS model structure. So we can, all right. Luckily, I've already run this. So hopefully this is working on your computer. I don't know why it's not working now, but it was earlier. So that should make this MixSIAR model text file. So this is the actual model that is going to be run in JAGS, and it's useful to save and also to look at. If you're submitting a paper, I would suggest submitting this because it is the exact, this is the model structure that you are fitting. So at the top, there's this section of a hierarchical fitting of the source means, fitting of the global proportion, the regional offset, the pack offset, and then for each individual wolf, it's calculating in ILR space. It's that regression with the global intercept plus an offset from region and pack. And then there's this math to back transform from ILR space to proportion space. Also to back transform the ILR offsets for the regions and the packs to what actually would make sense to us, which is a proportion region and pack. And then there's the calculation of the expected consumer isotope values. And here we have the likelihood definition. So the MixSIAR model, the JAGS model file is worth looking at before running the model. And then the last thing we need to do is to define the MCMC parameters. So how long are we going to run this for? I suggest that it's a good idea just to run a really short one first to check if everything's loaded correctly and the model is specified correctly. So I have this test option, which is just, I think, 1,000. So we can run that. And I'm having issues for some reason, even though this was working several times earlier today. Yeah. Sorry, that is because I didn't change into the correct folder. Sorry, bear with me. All right, so now it works. So this is now JAGS running that MixSIAR model.txt file, and the test will take a few seconds to run. That is not actually going to get a converged model. So you'll have to use a much longer run that may take 30 or 60 minutes in order to converge. And we can see that this test model is not converged if we look at the diagnostics. So let's look at the diagnostics. So this is not a course on MCMC, but so now we're getting posterior estimates of all the parameters and a few warning messages that I think we can safely ignore. And then we get these diagnostics, both the Gelman-Rubin and the Guicki. So this looks terrible. These should be like 1.00, 1.01, and they're at 15. So that's because we run our MCMC run was way too short. So yeah, we get the diagnostics, and then we get summary statistics for each of the model parameters that we're interested in. We have the mean, standard deviation, credible intervals, and then you also have access to the full all the MCMC draws for the full posterior that you can look at. The JAGS.1 object that comes at a run model is an RJAGS object. So any tutorial or any code that you have for processing those MCMC chains, you can use after the fact. So let's see. But the main results are these posterior density plots. So we have this was a really so this was you can see this for region 2, the estimate for marine mammals in green is bimodal. So that's really not very good. Also for the deer, it's bimodal. If we run the model a lot longer, then the model will converge and you hopefully won't have those issues. So let's I can load a longer run that I already saved. And then I can generate that output again. And then everything should look a bit better. So if you had run the normal MCMC, the longer run, then our Gelman-Rubins are more like 1.09 at worst, which is close to convergence, not quite, but close to convergence. And then you can reliably look at your posterior estimates. So for region 2, now that bimodality is gone, and we have a better looking posterior with credible interval. All right, so that's the wolves example. One thing that people often ask about is how to. It's hard to write a package that will give generally good looking output for in every case. So people will often want to modify the output for making nicer figures. And so I have a vignette that's specifically tailored to that. So these are not on Cranient, so you need the GitHub version. But let's assume that you had run the wolves example. We can add this return object equals true, so that will give you the ggplot as ggplots as objects that you can then modify. So here's our summary statistics table for each of the parameters. So we can look at that. You can look at for individual parameters that you're interested in, individual proportions. You can access them by using the row names of this saved data frame. So let's say we're interested in the proportion of salmon in the diet of pack 4 wolves. We can look up just in the DF stats data frame, we can look up just the pack 4 salmon estimate. We can find, say, for region 1 wolves, deer. We can find just the 2.5 and 97.5 quantiles. That's our 95% credible interval. And then we also have access, like I said, this JAGS one is a RJAGS object. So you can, if you look in the bugs output SIMS list, and then you refer to how the JAGS model file had named the parameters, then you can see that you get the same answer. So then that gives you confidence that this p.fac1, 11 is the same thing as the region 1 deer. So that's a good check. And then you can do things like, because we have the full posterior for each of these proportion estimates, we can calculate the say the probability that the region 2 wolves are eating more than 70% deer, say. So you can use the raw MCMC chains to do that calculation. This is just an example. So the probability that that proportion is above 70% is 22%. You can do things like calculate the probability that pack 4 eats more deer than pack 7. So you can get the estimates for pack 4 deer and pack 7 deer. And then you can look for each of the MCMC draws. You can that'll give you you can see whether it's greater or less than pack 4 versus pack 7. So that's what this is doing. So there's a 92% probability that pack 4 is eating more deer than pack 7, and you can visualize this by, say, making a histogram using the So this is a histogram of for each MCMC draw, you're looking at what is the difference between the pack 4 deer proportion and the pack 7 deer proportion. And then we can make a histogram. So we have a posterior of functions of the parameters. So that can be something of interest. And then another thing, if there's relatively small changes that you want to make to the output plots, you can simply modify those ggplot objects directly. So using this, saving the output of the output posteriors function, this will plot the overall proportion. Say if you don't know that a wolf is in a specific pack or a specific region, then this would be your best estimate of its diet. So you can do things like change the color. You can move the legend around. Any modification you could use ggplot for, you can modify the output without having to access the MCMC chains directly. So that's just a simple example of changing the fill and the color scales. And then let's see. Yeah, the other thing I was going to show is the continuous effect. So I'm not going to walk through the alligator example. There's a link to it here. That one takes a while to run, but you can, in that one, there's an example where the legend is on top of the nice area of the figure, and you just want to move the legend position. So that's a simple thing to do if you can get access directly to those ggplot objects, and then just say theme legend position right, for example, or you can change the color scale. But I'm sorry, I think I'm running out of steam. I think I should stop now and ask if people are following along or have questions. Yeah, so thanks for following, and hopefully that made sense.
Thank you, Brian. So that was a great presentation. If anybody has questions, please post it on poll everywhere. pollf.com, FE 2023. I can see there are some questions here. So I'm going to start relaying them to Brian, but you can go there and also you can upvote the questions that are already there, or you can post your own. So the first question, Brian, here is the person saying, well, although it's not ideal, if I need individual diets for individuals that are different each year, no repeated sampling, what is the best way to go about it? Would it be animal ID as a random effect nested within year? Would I still get reasonable confidence intervals at the individual level?
That is what I would suggest doing. That's a good idea. There's a limitation built into MixSIAR to only include 2 random effects. So if you have only one other random effect of interest, like year, then you can include individual ID as a random effect. And that's good. Good way to approach that. If you have individual, so you have non-repeated sampling, which means so you have 1 point for each of those random effects, the individual random effects. Then it's probably a good idea to not use the residual error term, because sometimes that can cause issues if you effectively have a random effect for each data point. It's best to use the process error only and turn off residual error, which I think you'll see documented in the manual or previous papers somewhere. That's what I would suggest, is to use individual ID as a random effect and process error only.
Mm-hmm. Great. So, so the other question is wondering if you have any advice on navigating data sets where sampling occurred multiple times and species were caught during different seasons. So an example here, in a lake, prey fish species caught in April and May, and larger consumers, predator fish, sampled in late summer, July. So how will you go about accounting for seasonal variability?
Sorry, was it that the
Yeah, it seems like,
is that the sources and the consumers weren't sampled at the same time, or is it that I'm not sure. I think you'd have to think as an ecologist about what so it's these mixing models they're assuming that the consumer tissue is comprised of the values, like the source values that you're putting into the model, with they get incorporated into the consumer tissue, and then you sample the consumer tissue. So you think about the turnover time of the tissue as well. So it can get a little more complicated to think about, but some tissues, for example, could effectively integrate the diet over years, or it could be much faster, like if it's blood versus bone, or something like that. So it's not just when you take the samples, but it's when those isotopes get incorporated into the consumer tissue that you have sampled. If it's just a matter of there's variability in the consumers based on season and year, then you can include the fixed or random effects of that, or continuous effect potentially of date. But it's hard to give a general answer because it depends on the ecology of the system you're working in.
Mm-hmm. Mm-hmm. And then this is a general question. So what advice do you have for folks just starting out with little or no experience with mixing models? How should they go about learning about these models, how to interpret its results, and how to understand these models?
Yeah, these can get pretty complicated pretty quickly, and I think they can also, it's pretty easy to get nonsense as an answer. Um, so I would start simple. I would plot your data, and just by looking at the isospace plots, you have a sense of what the answer should be. You know, I mean, check that the mixture points are within the polygon. So the isospace plot that MixSIAR is giving you is adjusted by fractionation as well. So it could be that the fractionation, you know, there's so many things that could be happening. Plot the data. Start simple. I mean, you don't have to start with MixSIAR. You can go back to, I think that Don Phillips et al. 2014 paper on best practices in stable isotope mixing models is a good paper to start with. And then you also don't necessarily need to use a mixing model. You can use something like Cyber to estimate ellipses and differences between groups and things like that too.
Mm-hmm. Yeah, there's somebody here that's currently working on the Deepwater Horizon oil spill, how it affected sperm whales. So other than carbon and nitrogen isotope data, this person also has satellite tracking data, distance travel, diving profile. Could that person integrate this data as covariates in mixing models?
Yeah, definitely. You just have to, I mean, the raw satellite data, you're going to think about how to construct the most meaningful either group, like a categorical or a continuous effect, whether it's, based on depth or area of use. I mean, there's a lot of different ways you could take the raw satellite data and turn it into a covariate that you could put into one of these models.
So it would have to be a covariate that summarizes the movement data for that given individual. Is that kind of what you're thinking?
Yeah, I mean, I'm assuming that you don't have, yeah, yes, because it's not like you have an isotope value for every satellite location point for that animal, I'm assuming.
Yeah, so probably it has to be a individual level covariate, some summary of the tracking data.
Yeah.
Then let me see. So the person, this person is trying to run MixSIAR models with multiple isotopes. The person ran a model with carbon and nitrogen, and then another model with carbon, nitrogen, and hydrogen from fish analysis in Lake Michigan. Apparently the person had very similar output from both models, regardless if CNN or CNNH. So what is your opinion about including more isotopes? For example, in this case, 3, but maybe more. Can that benefit or can it improve the estimates?
I would think so, because you're adding more information to the model, as long as it's consistent. If the third isotope is telling a different story, then it could make your uncertainty increase. But I would guess that you would have lower uncertainty with more tracers. But I don't think it's a problem to present multiple models and to present multiple models and multiple results and say what is the consistent story. That's kind of what we did with the mantis shrimp analysis. We ran several models. We ran models with we ran models with uninformative priors and two types of informative priors, and basically to build the case that the interesting result was robust to the priors that we used or the type of model that you use. So if you get the same answer with only 2 isotopes versus 3, then that just means your results are more robust, I guess.
All right. Is there, so have you already used examples with many more isotopes, or what was the maximum number? And did you see any change from running with fewer isotopes or with more?
There's a couple examples that use compound-specific isotopes that are included. There's one, they're both from Galloway. I forget the first name. But they're from isopod. The isopod and the Cladostera examples include no, it's fatty acid. Sorry, it's not compound-specific stable isotopes. But there's also examples of people using element concentrations in soil science where you have can have like 20 tracers or more. And I think in general, more tracers is useful as long as they're telling a consistent story and as long as the assumptions of the model are met, so that for each of those tracers, it's harder to visualize once you get above 3 or so, because you can't tell that the mixture data are within the polygon. And it's not that if you have 20 tracers, the mixture data don't have to be within the range for each. Yeah, it's just harder to visualize when you have a lot, but in general, I think it's helpful.
Yeah, and so I guess this relates to another question here is, how do you judge the goodness of fit of the model? And if the model doesn't seem to be fitting the data very well, what are the sort of things that you would look at in terms of improving the model?
If you don't have if you have a lot of I think it's more difficult with a lot of tracers. If you have few tracers, you can look at the isospace plot and you have a sense of what the answer should be. If you can't do that, you can't visualize that as well, it's a little trickier. I mean, you should be able to plot the posterior predictive distribution, but I haven't given a function to do that. But I know Andrew Parnell's SimMemR package will plot the posterior predictive distribution for your data, which is useful.
Mm-hmm. Yeah, all right. So just a couple of more questions. The person saying, maybe I'll cover this, but would this type of sampling error cover situation where there's suspected fractionation in the samples in water, for example, in water isotopes in plants versus well data? Or I wonder if there's a good way to deal with this. So I'm trying to understand, it wasn't entirely clear to me what the person's, maybe it's when you add the error term in your model, maybe that's what the person is talking about.
Yeah, I mean, the best is to have experiment that where you measure the discrimination factor for each of those sources for specific to your consumer. If you don't have that and you have to use literature values, that's commonly done, but it's adding uncertainty to the model. So if it's feasible, then I would say, I mean, the best thing is to try to do an experiment to measure the trophic discrimination factor. If not, I think, forget what was there's a CIDR package, I believe, where it's like a hierarchical by taxonomy way to try to estimate discrimination factors that you could look into as well. But yeah, I agree that discrimination factors can be an issue, and the uncertainty in the discrimination factor is an issue with these models.
Mm-hmm. Yeah. And then the last question is, because we are almost out of time, is and the questions are out of order here, so I'm not entirely sure what it was related to, but it says, so that would be the same if we have contaminant data. We could add the contaminant concentration as a covariate. It would tell us how contaminant and diet covary. That would probably be better than switch from Bayesian to frequentist to then get how diet affects contaminant concentration, right?
I'm a little unclear. I think I'd have to have more of a background in the.
Yeah, I'm not entirely. All right, so the person is interested in how contaminant perhaps might affect diet.
Or yeah, the
Uh,
relationship between the contaminant
right.
and
But
diet.
but then it's saying switching from Bayesian to frequent is to then get how diet's affected by contaminant. And
I mean, I think you could use the results of a mixing model with the diet is estimated and then plot, say, the correlation between estimated diet proportion and contaminant. Unless you think that, well, yeah. Yeah,
So
or you could
you
include
could have,
contaminant as an effect in the model and show directly that the, yeah, I guess the question is whether the contaminant is influencing the isotope values. Like the mixing models are estimating diet based on isotope values, and then you want to know what the relationship is between contaminant and diet. Yeah, so I'm not sure whether it's better to include contaminant in the mixing model or to take the diet estimate and relate that to contaminant afterwards.
so what you're like this last scenario that you mentioned would be you would allow maybe diet to vary from site to site or region to region. So you would get an estimate of the diet for each region or site, and then you could relate that to contaminant concentration kind of. So the model wouldn't have contaminant as a covariate, but you would kind of try to relate it afterwards. That's kind of what you're saying, right?
I think you could do, yeah, my brain's not working really well, but I think you could try it either way. You could include in contaminant or site as an effect in the mixing model, and then look at the diet proportions as a function of that, as a function of contaminant.
Yeah. All right. So it's about 1:30. And I think this was very useful. The questions were really good as well. So Brian, thank you very much for the presentation. I think it was valuable. And the code that you made available, it's really helpful, and I think it's gonna be of great use to our community.